Hello, everyone. It's a great pleasure to announce this extraordinary seminar by Olaf Heidenreich. Just a few words about Olaf. He has been in the leukemia field for a very long time and has done very seminal work on the role of transcription factors, in particular fused transcription factors, to the generation and function in leukemia. About his CV, I learned he has done his PhD actually at the Max Planck Institute for experimental medicine with Fritz Eckstein. After this, he has moved on to several postdocs, I would say, one in La Jolla, then in Tübingen and Hannover. And after that, he actually had accepted to become a senior lecturer and group leader at the University of Newcastle. But now already for 7 or maybe already 8 years, 6 years, he is back on the continent and in the Netherlands at the Princess Máxima Research Institute. And we're very much looking forward to having him here. It was the reason or how we managed to get him. He had made the mistake at a dinner 2 months ago to mention that he would anyway come to Barcelona, actually to visit Pablo Menendez. And I told him, yeah, but then you come a little bit earlier and visit us as well. So thanks for doing this and your audience.
Markus, thank you very much. Also for the kind words and the introduction. Actually, so far I did not regret having this dinner with you. So the other thing is, I mean, throughout my time also in Newcastle and also then here then in the Netherlands, I have this reputation of constantly interrupting speakers during their presentation and asking questions on the slide. So I'd like to encourage you to do the same, because that makes it easier for me to address things and not trying to flip back and forth to find the right slide. Yeah. So if you have a question, don't be shy, shout. And if necessary, I can also repeat the question because I understand that this thing is also online. All right. And I shall not walk too far away from the microphone, I was told. So as Markus already said, I mean, I started my career in Göttingen at that time at the Max Planck Institute with Fritz. And at that time, we worked with catalytic RNAs, with hamate ribozymes. And for those who do not know that anymore, at those days, we believed everything what you nowadays believe of CRISPR, we believed of that about the catalytic RNAs. The cure of all ills, it was going to solve all kinds of problems. From there, we moved to the RNAi, and that's something which I will stick to a little bit also with the presentation. And then now CRISPR, as I said, nucleic acid-based approaches to modify things, to get a very specific handle into the hand to possibly tackle diseases and to provide some cure. It's an old concept, and history mirrors itself. Anyway, so let's get started then. So, and sticking now with this cure or a specific cure, that picture basically shows how Heracles is trying to kill the Hydra. The Hydra is a snake-like animal, and for every head you cut off, 2 new heads are growing back. So basically, that reflects the situation which we still experience, and I should say I'm a scientist, I'm not a clinician, just as a warning. What we still experience with quite a few of our patients, that when they relapse, it's getting worse and worse. And the way how we actually deal with that situation still, and actually not too unsuccessfully, is we're getting the big club out in form of chemotherapy. Anything which moves or anything which is proliferating is being hit. It would be, of course, much more elegant to move to another story, a later poem about the Ilias, where Paris kills the almost invincible Achilles by his only vulnerable spot by hitting it at the so-called Achilles heel. And that is a paradigm, you hear that always as that example for targeted therapies. So, I mean, just to show this again, I mean, this shall reflect with my modest artificial art talent, the leukemic the cell and the normal HSC, if you like, where the difference is the, in our case, in the pediatric space, usually a leukemic fusion. And what we're currently doing is that we hit, as I said, anything which is proliferating is being poisoned by agents which interfere with DNA replication or mitosis. And it would be so much more better if we were able to take advantage of our genetic differences and exploit them for a more targeted therapy, leaving the normal hematopoietic compartment intact. And fusion genes, of course, have another important advantage compared to many other mutations, which are currently more frequently targeted also by pharmaceutical industry. And that is that they're usually, at least in the field of leukemia, initiating events. That means this is usually the first step on the way towards a fully transformed leukemic cell. During progression of the disease, you change from a pre-leukemic state to a leukemic state by acquiring additional mutations or epigenetic events and then you end up with a fully developed, fully transformed leukemia. But nevertheless, because we have here an initiating event, that means all the leukemic cells which are existent in the patient will carry this initiating event. And all the experiments which we have done so far suggest that they are also strictly dependent on these, on continuous expression of these fusion genes. So in principle, we have here an ideal target for cancer-specific therapy. And in some way, we're trying to exploit that. We as a scientific community, and everybody probably has heard of all the BCR-ABL inhibitors, which are being used as an example. Although that is strictly not fusion gene specific, because you target anything which is an ABL, independent whether it's fused or not. And it's also usually not such a clean drug. You also have PDGF receptor, you have KIT, and there when you look at that, and also now how these drugs are being used, also in GIST, etc., that is again this old wisdom of drug discovery. A little bit of dirt or unspecificity doesn't hurt. All right. So just a quick introduction into the pediatric AML. I mean, I hope I'm not carrying colds to Newcastle here. I probably do. But AML of the children and of the adolescents is a very heterogeneous disease. The most frequent groups are the MLL rearrangements, or nowadays I have to learn KMT2A rearrangements. The second largest group are the core binding factors, and here in particular translocation 821, which gives rise to the RUNX1E2 or RUNX1T1 fusion protein. This is, strictly speaking, the most frequently found translocation, at least when you look into the northern and central European area. When you go to the Mediterranean, there actually you find this one more frequently. So currently, whenever it depends on which literature and which author, senior author you're looking at, the claim is either this is the most frequent one or that is the most frequent one. And all these different subtypes have, of course, also different clinical outcome. And again, our RUNX1T1 is behaving relatively well in terms of chemotherapeutic response. So we get now here cure rates of between 70 to 80%. And if we are now also including targeted moieties such as Mylotarg, which is an antibody drug conjugate targeting CD33, we are slowly approaching 85%, which is quite good. On the other hand, we have a couple of translocations such as, such as the NUP98s or some subtypes of the MLL rearrangements, the KMT2 arrangements, which are doing relatively poorly with 70 to 80% relapse rates. What is also quite interesting to see and to notice is that when we look, and this is here now a UMAP of different AML subtypes of RNA-seq data, bulk RNA-seq data, we see that the core binding factor leukemias clearly segregate from the bulk of the other leukemias. We have here the MLL rearrangements. Here we have a group of NUP98s and UBTF, etc. And when we then looked in more detail, I don't have this slide here now ready, I deleted that one, but what that shows is you basically that about these these AMLs run no, and these AMLs run an HOXA9 mice1 program. So they are actually fundamentally divided into two classes. That is also reflected then, for instance, with newer targets or newer targeting drugs, which we are currently using, like menin inhibitors, where one can show that these ones benefit from the menin inhibition and those ones don't. All right, but back to our little pet, the RUNX1-ETO. The RUNX1-ETO, or the translocation 821, was first described by the giant of fusion genes and chromosomal rearrangements, Janet Rowley, back in 1973. And when you look at the citation here, you see that these were the golden days when people simply got their work published and did not look after impact factors. So this is actually both this publication and the better-known publication about the BCR-ABL, the Philadelphia chromosome, were published in the same year by her, and she simply didn't bother whether it's a French journal or whether it was an American journal, and in that case it was the PNAS or so. Important thing was it was published and people did take notice. The translocation fuses the DNA binding domain of RUNX1, and RUNX1 is an essential transcriptional modulator of hematopoiesis. Fuses the DNA binding domain, the so-called RUNT homology domain, to the almost complete open reading frame of the RUNX1-T1 or ETO. And that then recruits via NCOR and SEN3A histone class 1s to this transcription factor and or this complex. And basically what you're doing here is you convert, and as I already said, essential transcriptional modulator into a constitutive repressor. And that has consequences also in terms of networks. I mean, you can then look how these fusion genes establish their own transcriptional networks. And just to highlight this, and I'm still struggling here with the wrong arrow, is, for instance, just as an example, together in collaboration with Connie Bonifer, at that time then in Birmingham, we also showed that, for instance, that RUNX1-ETO drives a program which is dependent on the repression of CBPs, which is dependent on PUAF4 or BRAIN3A, and also the AP1 complex. And what that already indicated is that such a fusion gene, such as RUNX1-ETO, must, via this network and in collaboration with other factors cooperating epigenetic or genetic events, must then drive this so-called hallmarks of cancer. And when we are looking into the function of this fusion protein, we are always surprised to see that basically almost everything shows up here. So, I mean, we started this whole work with asking, what is RUNX1-ETO doing in a normal human leukemic environment? What people have done before was that they took systems, human or particularly mouse cells, and overexpressed it. What we did is, at that time, we applied an siRNA approach. The work just came out or was just going to be published by Tom Tuchel. And we designed siRNAs right against the fusion site of the fusion transcript. And this siRNA then, in theory, should be able to degrade our fusion transcript and should not touch the co-expressed RUNX1 fusion transcript, wild-type transcript, nor should it attack the ETO, which I should also mention is not expressed in the hematopoietic system. And when we did that, we observed that when we did a knockdown, that we impaired the proliferation of the cell system, the Kasumi1 cells. That also went along with a very, very remarkable loss of coronary formation efficiency. always stands actually for a mismatch where we simply flipped two bases. That's our negative control. And also when we pre-treated Kasumis and did then transplantation into cells, into mice, we also observed an increase of survival. The problem here, however, is that we did these experiments by electroporation. That is something which is difficult then to expand to. And also, I mean, the idea is, of course, I mean, you have here something which is able to, where you can do a little bit of biology with. But the question is, can we also use this potentially, can we harness this towards a therapeutic approach? And here I had a very capable, well, they're all talented, aren't they? A PhD student with Hassan Issa, who developed an LNP particle, a lipid nanoparticle, by combining several lipids here. And the most important one here in that context is this Delin MC3DMA, which contains an ionizable nitrogen. So this is not a set, but a nitrogen flipped by 90 degrees. And that has a pKa of 6.2. The advantage there is that you can form these LNPs at an acidic pH together with your siRNA. Your siRNA is, because of the phosphates, negatively charged. Your lipid is positively charged. You get a very efficient loading of these LNPs. And then once the LNP has been formed, and that is usually done by this microfluidic system, once the LNP has been formed, you neutralize the pH, you lose the positive charge on the surface, and that again ensures that your LNP is not going to stick to any single membrane. All right, and this principle is, I think, well known. I think the vast majority of you have already benefited from that, because the very same principle has been used with slightly different ionizable lipids for the COVID vaccination. So, does it work in vivo? And here we are now using again a system where we try to be as pediatric as possible. We injected newborn mice with Kasumi1 cells. This is this A21 cell line. We let them engraft. We then treated the animals with these LMPs and first asked, do we get a knockdown in vivo? The so-called on-target activity. And what this shows here is, here again, we have this mismatch siRNA. In that case, we use a slightly chemically modified one. Here, the active siRNA. And you see each of these bands or reflects one single mouse. And what you see is that basically in the majority of the mice, we get a very significant knockdown of the fusion protein, demonstrating that there is this on-target activity. That also has an impact on survival. So this is shown here, where we get, with a temporary treatment, we get a modest, I think, increase in the survival. The interesting thing is, if you now take a material from here, from mice which has succumbed to the disease, be it from the mismatch or from the knockdown, and retransplant this into new mice, you get this picture. And this is without any further treatment. So what this shows is that material from here creates a much less aggressive disease compared to material from the mismatch treated groups. And this is a classical proof for that leukemic self-renewal is here long-lastingly impaired. So this was very encouraging, but this is all cell line gymnastics. So the question is then, what about primary cells? And there we had to overcome a couple of hurdles. Let's start with the LMPs. I mean, here we have a UMAP showing flow data from, or actually a TSNE, not a UMAP, a TSNE showing flow data from a panel of 12 different antibodies. And you see here, as an example, the CD34 positivity in these AML cells. When we do a knockdown with the siRNA, here we have this LMP. What we see is, or when we do a treatment, either with a mismatch or with the active siRNA, we don't see a difference in terms of phenotype. That was a bit disappointing. As I said, with the Kazumis, we had this very nice and strong phenotype, not with the primaries. So, and when we looked at that, one of the reasons was, if not the main reason, the knockdown was miserable compared to what we see in Kazumis. What to do? And so we consulted a little bit of literature, and what we wanted was a target which is expressed on basically as many hematopoietic cell types as possible, which is efficiently endocytosed, and which, no, the other way around. The advantage is we can go for something which is as much expressed in the hematopoietic system, but not in other systems, is to avoid or to minimize gobbling up by other tissues. We can afford to go for all the hematopoietic cells because the specificity comes not from the vehicle, but from the cargo. Remember, the siRNA is very specific for the fusion transcript. And well, what we finally came up with is the very late antigen 4. Again, as I said, this is expressed on virtually all hematopoietic cells and is also taken up by an endocytotic, usually a clathrin-mediated pathway. And that also either goes into, can go into a lysosomal pathway or can be recycled and show up again on the surface. So looked to me rather promising. And that was another big advantage, that very late antigen 4 has been already targeted in rheumatoid arthritis when people used these type of inhibitors to interfere of the recruitment of T cells to inflammatory sites. And this is simply a tripeptide, LDV, leucine, aspartate, and valine. And with the protection, with an N-terminal protection group, methylphenylurea group. What we also did is we included a lysine which carries on the epsilon group in a cyto group. And the reason for that was, and that is now worked by Laura Swart, another PhD student in our group, because we used now click chemistry to link this LDV to DSPE, so a polyethylene glycol lipid, which then was integrated into our LMPs by a process called post-insertion. So what you do is you simply have already your preformed LMP with the siRNA, and then you integrate this new lipid with the targeting moiety into your LMP. And the advantage there is also that you have automatically orientation. That means your target really shows, or your targeting moiety really shows to the outside. And that worked quite beautifully. Here we have now, again, the primary cells. Here we have DAPI staining for the nuclei. These are the primary cells with an untargeted LMP. And here we use an LMP which is labeled with a fluorescent dye, which integrates into the lipids, into the lipid layer. And that's how it looks like when we have the targeting moiety. So we get basically 100% of the cells stained. And when we go back to this example here with a failure, when we now take such a target lipid, again, when we look at the mismatch, no comparison to the mock-treated cells. However, when we now have the active siRNA, we get our desired and so badly needed phenotype. So we get a complete shift of populations, a loss of this central part of this very CD34 positive section, and a gain of CD34 negative populations, indicating ongoing differentiation. Pharmacokinetically, the question is, of course, do we reach the bone marrow? And this is just a quick detour here. So again, what we did is we injected either non-targeted LMPs or LDV-targeted LMPs. And here in that case, we did not use a lipid integrating dye, but we conjugated this dye by the same click chemistry and inserted in the same way as we did with the LDV, because we observed that we do lose fluorescence during these experiments. So that was more stable. And what we, to our delight, could see, first of all, both LMP types enter the bone marrow. So we can see them. These are here pictures with the femurs, actually through the femur. So the bone is still there and shields to a certain degree also the fluorescence. And when we then look 24 hours later, then you start seeing a difference between actually the targeted ones and the non-targeted ones, indicating that the retention by the targeted molecule is higher. And that is, again, is making sense, because the way towards the bone marrow should not be affected or hardly affected by the LDV, ideally. The LDV, the targeting moiety, should make a difference once it has reached its target tissue and should have a longer retention time there. And we could then also show by looking in, because this construct or this targeting moiety does not discriminate between murine and human VLA4, we could also show that by plotting here CD34 versus the fluorescence, that we get about, these are here the CD34 positive HSPCs in these two quadrants. And you can see that we get more than 2/3 of these HSCs or HSPCs being labeled by using these targeted LMPs. So there's a difference also compared to the non-targeted one. So that looked all good. However, so I mean, first we made a step back because we wanted to work on primary cells ex vivo to do some experiments there. And I will stay with that for the rest of the talk. So first of all, we needed a system which allowed us to look into the MSC, to look into the expansion or the culture of primary cells. Primary AMLs is difficult because they either go straight into differentiation, you lose all the immature LSCs, the leukemic stem cells, or they die. So what we did then is we developed a co-culture system, which is serum-free, which established, and this co-culture consists of mesenchymal stem cells or stromal cells, which we obtained from pediatric bone marrow. We co-culture them, and that allows us to expand AML cells, as shown here. This is at the time of seeding. This is 10 days later. And here you see examples on how these cells expand over time compared to AMLs alone in blue. And that also is not only that we in total expand the bulk, but we also expand an LSC-like population, which is the CD34 positive, CD38 negative, CD45RA positive population. That what Parash Vyas has described as LSCs, or LSC-enriched population. That is also growing. And we can monitor the fusion gene in such a culture for a period for sometimes over 3 months. However, at that time, we have lost the LSC population, I should say. Anyway, so we had now the tools at hand. And so we basically repeated these experiments, and this is now work mostly by Paulina Derbianco together again with Laura Smart. So we seeded our primary cells on these co-cultures, and we then treated them with the LDV LMPs, or and either with active or inactive siRNA, shown here again. We have a selected modification built here in, mostly on the pyrimidine nucleotides of the antisense strand. We included phosphorothioates to protect against exonucleases. And these siRNAs then lead now to a robust knockdown of these fusion proteins. And this is also then seen here on the RNA, where we have then mismatch active siRNA day 3. And when we do repeated doses, we see that the fusion protein completely disappears. And that does not only work for the RUNX1-ETO, but also works for other fusions. For instance, here we are targeting the MLL-AR4 just as a little sidestep. This is one of the translocations which has a relatively is associated with a relatively poor clinical prognosis. Here we're using actually a completely modified siRNA design, including a 5' vinyl phosphonate modification on the antisense strand. And here again, we see in these, in that case, in the cell line, a very nice knockdown of the fusion RNA and the fusion protein. We see an impaired mRNA expression also in primary, or in that case, PDX cells, impaired proliferation, and also the clonogenicity breaks down, depending on whether we have targeted on unmodified construct here. So, I mean, the important thing is it works not only for the siRNA, for RUNX1-ETO. And please also note that here in that case, actually, we used the siRNA against RUNX1-ETO as a negative control, again, highlighting the specificity of the approach. So back to RUNX1-ETO. So what does it do in the primaries? So, and this is now work together with Sophie Callaway. So the first thing what she did is she looked into ATAC-seq data and combined this with RNA-seq. Here we have then basically the comparison between, again, our mismatch control, our active siRNA. Here's the differential gene expression. And we see that there we have a gain in where we have increased gene expression, that we also have a gain in binding sites, vice versa. Loss is associated with loss of binding sites here. And when we compare these gained or lost binding sites against AP1 or CBP binding sites motifs, we see that the CBP motifs enrich in the gained sites. AP1 sites, which usually are associated with proliferation, are in the lost sites. Also, when we compare the different patterns, we see that we are losing an HSC-like chromatin pattern, and we are gaining a GMP or monocytic and neutrophilic chromatin pattern indicating changes in the chromatin accessibility. So again, these ATAC-seq data show that we have a change towards a more differentiated phenotype. And that is also seen when we look at distinct or single gene transcription factors. Here we're looking first, we're looking at CBP epsilon. That is the member of the CBP family, which shows the strongest induction upon RUNX1-ETO knockdown. Much stronger than, for instance, what we see with the CBP alpha. And when we then again go back into the chromatin, etc., here we have combined Kasumi-1 data, which we have generated with Connie Bonifer before, together with ATAC-seq and RNA-seq data from our PDX. And what we can show is that in both in the cell lines, we see at a region of about 5 kb, 5.5 kb upstream, downstream of CBP epsilon gene, we see, or of the locus, we see an increased binding, increased opening. This is also reflected in the primary cells. We see also a loss of RUNX1-ETO binding. So RUNX1-ETO is sitting there, and loss of RUNX1-ETO binding is associated with opening of the chromatin structure. And that translates then also here as an example for the PDX, same with the Kasumis, we see a massively increased CBP epsilon transcript expression. What Polina then also did is she did single cell RNA sequencing on these samples. And the very first question was, here we are using one PDX and two primary patients. And here we also have now cells in there that do not express the fusion transcript, that are wild type, that are normal. And that can be simply identified by the expression of ETO or RUNX1T1. I already mentioned that RUNX1 ETO is usually not expressed in the hematopoietic system. So we use that as a cutoff, and logically, of course, the PDX doesn't contain any normal human hematopoietic cells anymore. That is lost. When we looked at those cells, just to start with, here is a classification or a mapping of the primary cells which we have, either with a mismatch or the active siRNA. Here in red is indicated cells I obtained from the RUNX1 ETO treatment, the knockdown, or the mismatch in blue. And what we basically see is that in these normal hematopoietic cells, we don't see any sort of changes. And that goes across all the different subtypes, including early HSPCs, as shown, for instance, here in the blue ones. For the RUNX-ETO positive cells, it looks quite different. And I'm starting here with a PCA. And the reason for that is usually a PCA. If you see a difference in the PCA, then you know there is a difference, because there you look at a linear distance. With a UMAP, this is different. I mean, there you sometimes get very massive changes. And when you then really look in a PCA, you hardly have any difference at all. But what this shows actually is that we get, blue is again the mismatch control, that the RUNX1 ETO knockdown leads to a bidirectional movement of the populations. That is also shown here in the pseudotime analysis, where you see that here the central core is moving to the left and the right. Looking at some transcription factors again, and here returning to the CBP epsilon. The CBP epsilon actually, or the CBP epsilon positive, particularly with the highly expressing cells, love to move to the right. Hardly anything on the left side. Then we looked at markers such as the IL-5 receptor, again, left side, and this is actually a marker towards eosinophilic differentiation. So that was something which we found a bit surprising, that the CBP epsilon seems to be more on this side. And also when we looked at GATA2, GATA2 accumulates again on the left side. GATA2 has been described before also being part of a stem cell associated heptate of transcription factors. But here it goes clearly, the higher the expression, the more to the left side it moves. And looking then at a couple of differentiation markers, such as the resistine, just as the resistine or the SERPIN1A, which indicates granulocytic differentiation, or then the RNase 2, which indicates eosinophilic differentiation. This again suggests that we have here this bidirectional differentiation going on. And this in combination with CD24, which is a don't eat me signal, which protects the more, amongst others, the more progressed, differentiated myeloid cells against killing. Again, we see here that the extremes, which are also shown here again, these are the ones which are CD24 positive. The other ones here in the center are not. So that if you put this all together, I already mentioned that a couple of times, is what that shows is that this bidirectional differentiation goes on the one side into the granulocytic direction, whereas on the other side, we also have now an eosinophilic differentiation going on. And that is insofar interesting because eosinophilic and also then there with linked to basophilic and mast cell differentiation has been recently described as one of the troublemakers when it comes down to MRD detection, minimal residual disease detection, by looking at the fusion gene. Because longer living cells of the eosinophils and of the mast cells tend to stay around. And if they're generated from these leukemic cells, they give you a false positive signal for RUNX1E2 positive cells. Actually, it's not false positive, but these cells are not able to establish a leukemia anymore. They have passed the point of no return, but nevertheless, they give the signal. For that reason, MRD is problematic for these types of leukemia. And that is, for the interest of time, just highlighting that when you look at the composition of these different granules, this is here typical granulocytic granule with which is the corresponding genes are expressed on the left side of the granulocytic differentiation part with azoruridine, cathepsin G, MPO, etc., elastase. Whereas on the other side, we have then again the RNase 2, 3s. We have several markers, and again we have here the CBP epsilon, which seems to be one of the drivers of this type of differentiation. That's something which we're currently looking into it. And just to finalize this whole thing, I do have 2 or 3 more minutes. This is actually now here a single cell map by Andy Zhang, which has been around already for quite some time in bioRxiv, but which has been also been officially seen the light of the day in blood cancer discovery. And Andy very kindly shared all the data with us, quite a bit of an advance of that. And what you can see here, and what I would like to highlight to start with, here we have the area of the more immature LMPP, MPP, and HSC cells. Here we have the granulocytic differentiation. Remember, we can't see granulocytes in aspirates because they're being removed by the Ficol purification. And down here, the monocytic differentiation. Up here, close to the erythroid and megakaryocytic group, we have then the eosinophils, the basophils, and the mast cells, or their progenitors. So when we now plot our control, our mismatch control onto that, we see this picture where we have cells here in this immature area. We have cells in the late GMP section, indicating again granulocytic differentiation, so basically reflecting what I said, the left side of the differentiation. And we have here another group which already shows an indication towards the eosinophilic basophilic differentiation in mast cells. When we now look at the mismatch, of the active siRNA, there we get that. Basically, we're losing this complete middle point. We're ending up with something which is quite advanced in differentiation towards granulocytes and also quite advanced in differentiation towards the eosinophils. So we're losing our, what I would consider LSC leukemic stem cell enriched populations. And one of the questions, of course, I mean, we're using here ex vivo co-culture conditions. We have cytokines in there, we have inhibitors in there. Actually, we have 2 strong inhibitors against differentiation. For those of who may know that, we're using stemreganin, which blocks the aryl hydrogen receptor, and we're using UM171, which it blocks the co-rest activity. So it's very difficult actually to overcome that. But when we do this with the siRNA, this should be actually, when we do this with siRNA, obviously we overcome that. So loss of RUNX1E2 and the cells are not being blocked by these 2 inhibitors anymore. And I don't know whether you can see that. Anyway, so you should still see somewhere this frame that should be here in light gray. But I think you still can imagine or appreciate these are now primary cell data from Zander Lambo's cancer cell paper of A21s. And we also did this with data from Sophie Callaway. What you see is, again, I mean, if you already take the fresh AML sample, you see already this bifurcated, this bidirectional differentiation going on. So I don't think it's an artifact of our conditions. It's something which you see in the patient directly. And that, I think, strongly indicates that we're losing these immature cell populations. You can also, going back to this horseshoe-like presentation of the Polina, you also see that here again, we have here the CD34 positive populations, and they're basically gone when we do this knockout, where the knockdown of RUNX1E2. And also when you believe, should you believe in LSC scores, like the LSC6 score, again, you see the same thing. You're losing the majority of the cells which express or which harbor this score, or which score highly on that one. And also looking at the already mentioned CD34 positive, CD38 negative, and CD45RA positive populations, they are basically to a large degree eliminated with a knockdown. And then when we do limiting dilution in PDX, again, what we do see is that we see a reduced ability of those populations to restore growth in 96 well formats. So that, I mean, this is not a complete knockout or knockdown, but I mean, here in that case, you also possibly have limits in reaching this so-called point of no return. So probably requiring longer persistent treatment. And as I said, I mean, our conditions do not favor differentiation. And with that, just 5 minutes over time, I'd like to come to an end. So what I try to show is that these leukemic fusion genes, that they can be efficiently silenced in primary AML when you use these LMP siRNA constructs. And that also means you have a tool to ask other questions in primary cells. For instance, now, I mean, knocking down what happens with CBP epsilon as an example. What happens if I take certain other members out of the game? So that is now feasible. We show that the fusion, that this RUNX1E2 fusion, the loss of RUNX1E2 fusion drives this differentiation, and that it goes into two different directions. But that this leukemia already has inherently this tendency of going into both directions. And I think particularly with the eosinophils, that was a bit underappreciated. I should also highlight this eosinophil differentiation happens in the complete absence of IL-5. We don't have IL-5 in the system. We don't have cells which produce IL-5 there. Yeah? And nevertheless, they go down that route. And that is a bit surprising. Well, that's shown again that the silencing of leukemic fusion genes also in primary cells impairs the leukemic self-renewal. And just following this credo of mine that they might be actionable targets for therapy. And what we are going to do next is, besides looking into CBP epsilon, which was actually requested by one of the reviewers, is look into the in vivo efficacy now of these particles in PDX. Again, coming back to this question of what is the point of no return? How far does the differentiation need to go to completely eliminate self-renewal? And how to bring it into the clinic, particularly which patients do you want to use? How many patients are out there where you can test that? Which schedule, after induction, during induction, where do you want to do that? Or just in maintenance? And last but not least, because we are not talking about thousands of patients, who would be the interested commercial partner with that? And that is the biggest hurdle, I think. And with that, I would like to thank all the people who contribute to the work. I think most of the people who are in FAT here are already highlighted, the funders. And I'd like to thank you for your patience. And I do hope for the one or the other question. Thank you.
Okay, thank you very much. I think we have ample time for questions. We start, of course, with PhD students. Not anymore, PhDs, sorry.
Hi, thanks for this presentation. Very nice work in primary cells. So I was curious about this co-culture of primary AML PDX with these stromal cells. So after this expansion, have you tried to like transplant them back into mice and see if it's the latency changes? And also if you check if there's some acquired mutations during this expansion time. Thank you.
I mean, this is a very good question, actually. So first of all, yes, we can retransplant. So these cells show still keep retransplantation potential within a fortnight. And in the case of the mismatch, also after this relatively intensive LMP treatment. And in the first set of experiments, it looks like as if the mismatch, what we did is actually we transplanted the mice. And then because we weren't too patient, I'm afraid, we killed them and looked into the femurs after 1 month. So we detect in a couple of mice of the mismatch as treated leukemia, which re-engrafted there. We did not see anything in the RUNX1E2 knockdown group. Yeah. But as I said, we killed them, unfortunately, too early, and we need to increase the numbers there. But currently, it's a black and white. But that may change. I don't know.
Thank you.
See?
Thank you. This was beautiful work. I was wondering, how long do you need to lose the fusion transcript for in order for the cells to like really get lost? And do you expect then that you have to retreat the patients for a couple of times, or is just one treatment enough for the cells to disappear or differentiate?
Currently, we're doing in the primaries, also for the retransplantation, we're doing 2 to 3 treatments with a 3-day interval. And again, I don't know whether 3 treatments will be sufficient to completely eliminate self-renewal potential in vivo. I mean, there is quite a bit of literature coming from also from the monocytic side, but also from the eosinophilic side, where they showed for other model systems that they get an aberrant differentiation and that these guys retain re-engraftment or engraftment potential and can restore leukemia. As I said, I mean, our current experiments suggest that this might not be the case here, but the numbers are small. We're talking about 5 mice each. So I think it needs a bit more of attention from our side then.
Hello, so nice talk. I have a kind of technical question about the LPS, lipid nanoparticles. How good is the targeting towards the cells, towards the AML cells? Have you checked if the nanoparticles go to other organs? For instance, I heard that they have tropism towards liver. Have you found this?
They have a strong tropism to the liver as well. And again, I mean, what the targeting is doing, as I said, it affects the retention, but it will not affect the primary entering of a tissue. And the liver is still a massive challenge for the LMPs. So actually, I think more than 50 or 60% of our LMPs end up in the liver. I should also mention, I mean, the very late antigen is also to a certain degree, not as highly, but it is expressed by the Kupffer cells. Yeah? So they also probably feel addressed there. We are currently trying to address this issue by selecting LMP formulations with an increased inherent tropism for the bone marrow. And because I think the clue will be the formulation itself. The targeting will add, again, I'm repeating myself, the targeting adds to the retention. But to reaching a tissue and avoiding another tissue, that is not the function of a targeting moiety. So we need to do something with the lipids.
Yeah. So thank you. In the case, so in your case, that you mentioned that about 50% of the lipid nanoparticles go to the liver. Have you observed toxicity in the liver?
When we look at the common parameters, that is weight, etc., and also alt-AST, we don't see that. And we also don't see a significant increase in, for instance, inflammatory cytokines, etc. But again, that may change with the next formulation, which we're going to use. Yeah.
Hello. Well, thanks a lot for the talk. I have a naive question. So does RANX1 activate or repress? Is it an activator or a repressor? Because you've shown in the beginning the partners of ITO, and they seem like repressors.
Mm-hmm.
So does it repress differentiation genes, or does it activate leukemic stem cell genes?
That's a very good question. To start with, I mean, again, by its very nature, it's primarily a repressor. So if you, for instance, create RANX1 binding sites in a given promoter, or RANX1 binding sites, and then you ectopically express this fusion protein. These are very old data back to Stephen Neimer and before that. You can repress the gene. You will repress it. It's more complicated. Also, I mean, when you look at the very first responses, for instance, when you use a DTAC or something like that, you first see in the first wave that genes are de-repressed. The second wave, then, there you have then also genes which show a mixed phenotype. Some go down, some come up. And that indicates, I mean, you could interpret this in a very simple way and say, okay, this is a completely different set of transcription factors. And they now, because they're de-repressed, they will activate the other genes. This, or will repress the other genes. It depends a little bit. So, I mean, the question is, can RANX1 act as an activator somehow in a certain context? Yes or no? And the data which we have is that in the second wave, you have genes which upon RANX1 go down, like cyclin D2 or CDK6. Other people have also seen that. And we also have very strong binding of RANX1 at those loci, or at elements upstream of that, which are known. And also when we do high C, also we know that they're interacting. And the question is then, are these then classical enhancers? Are these more silencing elements, etc.? So that by silencing the silencer and together with an activating signal, you then create this activation of such a factor. That's the more complex. I mean, it runs against Occam's scissors. I freely admit that. But the fact is, we do have the binding there, and the modulation of that binding is associated with the response.
Thank you.
Okay, we have time for one last question. Then I'm happy to take it myself. So do you think this point of no return, which is of course extremely exciting, this is what we want to achieve in therapy, would you expect this point to be identical for all fusion protein-driven leukemias or very different?
In principle, I would expect it should be at least similar. However, I mean, being here in an area where Thomas Graf basically reprogrammed virtually anything, I should be a bit more cautious about that. So I would not exclude a context-dependent effect. But on the other hand, I mean, usually you don't express CBP alpha very highly in those tissues. So I would expect a similar end of a point of no return there.
Okay, then please join me in thanking Olaf once more for his presentation.